Dr. Jeanne Marrazzo is Professor of Medicine and Director of the Division of Infectious Diseases at the University of Alabama in Birmingham. She is internationally recognized for her research and education efforts in the field of STIs, especially for those areas of the field which affect women's health. She's a Fellow of the American College of Physicians and the Infectious Disease Society of America. She's also the Chair of the American Board of Internal Medicine Council and also chairs that council's Infectious Disease Specialty Board. She's board certified in infectious diseases and she loves to conduct her research on the microbiome. So I'm going to leave it there and save any more embarrassment for Jeanne. Please come up and give us the lecture. Thank you.
What a wonderful way to start my birthday. Thank you. I was trying to do it on the down low, but clearly that was not meant to be. Charlotte Gatos, thank you very much for letting everybody know it is a good birthday. It's a great place to be here. So I could not be more honored and more humbled to be part of this all-girl plenary session, I want to point out. I have two fantastic speakers succeeding me, and I'm going to update you on the vaginal microbiome somewhat selectively. Actually, it was in London when the organizers kindly invited me to do a plenary, and at that plenary I talked about the new molecular methods for defining vaginal microbiome. That was very new at that time, and we have come a really long way. So let me start by trying to take you through some of this, and I'll explain the selection of the content in a moment. These are my disclosures. So what's new and what am I not going to talk about? Okay, so what I'm not really going to talk about very much is treatment of BV. And for that, I would refer you to the presentation on the new Lancet Commission on STDs, which is going to be presented today from 12:30 to 1. Kat Bradshaw is actually going to present the part on the bacterial vaginosis section that Jane Schwebke and she and I put together, and that really is a call for new approaches to treatment and managing the vaginal environment. So I encourage you to take a look at that. I'll also point out that there are a couple of trials going on. There's a trial that just finished looking at a novel biofilm disruptor that I ran along with Jane Schwebke and Julie Dombrowski in Seattle. Results should be coming in the next 6 months or so that's exciting. I know we're not supposed to use the lactobacillus in yogurt to treat vaginal problems, but I couldn't resist this lovely image. And it's actually from a great article in BBC Focus, Science Focus magazine, that interviewed a number of people in this room and talks about the promise of probiotic treatment for BV, which, as you may know, is being assessed in a trial led by Craig Cohen right now using the lactin V, Lactobacillus crispatus. So these are things that we can anticipate in the coming months to years, but they're not ready for prime time yet. So take a look at the information of those on clinicaltrials.gov. And since the last time we were together, we have had the first approval of a nucleic acid-based amplification BV-associated bacteria diagnostic test, namely the BD-MAX vaginal panel, that has a very sophisticated algorithm for detecting quantities of Lactobacillus crispatus as well as BV-associated bacteria. So very exciting, and a lot of presentations on that that you should be hearing about today. So stay tuned. But I'm not really going to talk about those things because, unfortunately, and that's what the Lancet piece really emphasizes, we don't have a lot that's new in treatment of BV. And that is what is so challenging basically, about this problem. So what am I going to talk about? I'm going to sort of revisit what a healthy vaginal microbiome is, because I think we do have more data to understand that. And I'm going to focus on the consequences of not having it, particularly with regard to the increased risk of HIV acquisition. I loved Kevin DeCock's plenary last night. I think his comments about convergence and divergence of STI and HIV set the stage perfectly for this, because I've always felt like, HIV was really driving the wave, riding the wave. STIs were towed behind this little life preserver. Now, between the emergence of STIs in the era of PrEP, plus some of the data I'm going to show you on vaginal microbiome and HIV acquisition, I think we really need to be up there on the wave with HIV, and that's a great metaphor considering where we're at. So I'm mostly going to talk about that, and some of this is data that either hasn't been published yet or was literally just published in the last month, and I think if you haven't been going to some of the HIV meetings, you may not have heard about it. So I'll touch on that. And then I'll just close by thinking about some ways that we might start innovating in our approach to maintaining the normal vaginal microbiome and defining pathogenesis, which we have really started to do. So just a reminder, I know not everybody thinks about the vaginal microbiome 24 hours a day like I do. In fact, I can't think of anything I'd rather be doing on my birthday than giving a plenary on the vaginal microbiome. Remember that there are many benefits to the optimal vaginal environment, and I'll talk about that in a moment. The main things are that you have protections from pathogens and you don't have vaginal symptoms that are referent to bacterial vaginosis. So you're less likely to get BV, HIV, chlamydia, gonorrhea, trichomonas, and there are really great prospective data, even from the cultivation era, to show that. You're also, you're less likely to transmit HIV, that should be transmitted, and if you have HIV, you probably have lower levels of genital HIV. The main reason that people originally got started looking at BV was because of the pregnancy-related outcomes, and that before HIV, those were really the big things that people thought about. And we know that there are now optimal birth outcomes associated with a lactobacillus dominant microflora, and that would be Crispatus or Jensenii, that are not just short-term, meaning fewer pregnancy-associated infections, normal birth weight, normal timing of delivery, so fewer preterm deliveries, but also long-term. And this is data that has really blossomed. The field has really blossomed since the last time we talked about this. For example, we now know that infants born through a healthy vaginal canal, particularly as it relates to C-section born infants, are more likely to have lower rates of autoimmune disease. I think the data are better for asthma than anything else. And we also now have really interesting data to say that as soon as a baby is delivered through a healthy vaginal microbiome, there's immediate colonization of the baby's oral cavity as well as the gastrointestinal tract and the genital tract with the mom's healthy vaginal lactobacilli. I can think of no more intriguing example of this than Gloria Dominguez-Bella's lovely, small, but really intriguing study that was published in Nature Medicine just last year. And this follows on her study in PNAS a couple of years ago that showed that delivery mode shapes the initial microbiome of the infant. So if you're born by C-section, what are you initially colonized with? In your gut as well as your skin? You're colonized with skin flora, largely staph, Corynebacterium, diphtheria, that sort of thing. If you come through the vaginal canal, you're colonized with Lactobacillus species, or the species of BV if you've got a microbiome disruption. So that was the first sort of proof that really kind of established, I think, definitively that that was the way that you are initially colonized, and really important for immune modeling. That's probably why these long-term outcomes are so critical. What she did then was this really intriguing study where she took 18 infants and their mothers. There were 7 born vaginally and 11 delivered by scheduled C-section. And what she did with 4 of the C-section baby-infant sort of mother pairs was to colonize the babies born by C-section with the vaginal secretions of the mom, assuming the mom had healthy vaginal secretions, and showed that immediately those babies were colonized throughout their body with the helpful or healthy vaginal lactobacilli relative to the C-section babies. The prevalence of C-section in the United States is about 32%. I just saw the updated statistics from CDC or whatever. So you can imagine that this is potentially a huge impact on the baby's immune system and as they age. And you know we're having a lot more problems with asthma and allergic mediated issues. So fascinating data, just a reminder when we come back at the end of the talk to thinking about innovative changes in the vaginal microbiome. Now for those of you again who need a little refresher, remember the spectrum of the vaginal microbiome that I've already alluded to was defined very early on, largely with Graham stain and cultivation, and on the left there you can see the beautiful Lactobacillus predominant, which would be a Nugent score of 0, and on the right that anaerobe predominance with clue cells and a mix of anaerobes. Since the time that we've met in London and elsewhere, use of molecular analyses to define this environment has really pretty much become the standard. And in general, what I think is intriguing is that they pretty much continue to complement the traditional methods. If you look at what we found out from the molecular analysis, and Sharon Hillier would be delighted to hear me say this, there was an initial surge, I think, of excitement. Maybe we would find the pathogen associated with BV. I don't think we've really done that. We've certainly clarified and detailed the community, but I don't think it's been quite the holy grail that we thought it was originally. I'm also going to talk a little bit about newer methodologies, mostly proteomics, to define some aspects of the microbiome. The bottom line is that all of these methods are defining a spectrum from what I will call optimal to less optimal. I started even thinking about not using the term BV because, as I'll talk to you about in a second, I think that BV is a simplistic and reductive term, and I think we're talking about different syndromes or different actually microbial communities that probably impact different local inflammatory responses, and I'll come back to that in a second. But the bottom line is that in the optimal vaginal microbiome, and I define optimal as healthy birth outcomes, lower rates of infection, whether you're talking about it in the reproductive tract or acquisition of infections, that's an optimal lactobacillus, Jansenii or crispatus, pH acidic environment. That's really what it is. This also gets you away from talking about, well, if 70% of women in Africa have BV, isn't that normal? That may be the norm, but it's not optimal when you talk about acquisition or risks conferred by that environment. So it gets you away from that sort of more, I think, culturally very important thing to think about. What's normal, what's the norm, and is that the best thing for the person involved? Okay? This is just a paper I'll use to introduce this concept of diversity of the vaginal microbiome, and beautiful work by Sujatha Srinivasan in David Frederick's lab. Sujatha's here. Always like to present this, and this is very similar to what Jacques Ravel and others have shown. So you could really choose papers with any of these groups and you get a very similar thing. And what you're looking at here is 16S ribosomal DNA, PCR, and PIRO sequencing of a couple hundred women. Each of these lines of colors is an individual woman, and you can see the AMSL score is under the letter A, the Nugent score is under the letter N. If it's red, it's abnormal. If it's green, it's normal. And essentially what you're seeing is that there is sort of a bimodal split. Everybody down below who is supposed to be normal clinically by AMSL and Nugent seems to be predominated by those two colors, orange and red. Those are Lactobacillus crispatus and our very confusing friend Lactobacillus inners, which I'd like to come back to because we really don't know what Lactobacillus inners is doing. It doesn't make lactic acid, it doesn't make the vagina acidic, and yet there's a lot of it around, and in fact you can be completely quote-unquote normal, at least by our crude measures of AMSL and Nugent. What I think is even more interesting, if you look at the top, the women who have BV defined by those criteria, you've got some clearly distinct groups. You can see the women on the top are dominated largely by that green bug, which is Sneathia leptotrichia. You may remember that that has been associated with urethritis in males. So you may wonder, are those a group of women who have acquired sexually associated BV because their male partners are continually exposing them to Sneathia and leptotrichia? Absolutely. And then you can look down and see the pink-dominated people. Those are Gardnerella vaginalis, and et cetera. You can sort of keep going and looking that there are really diverse heterogeneous communities. So I think, again, this is only 200 women. It's a selected sample from the Seattle STD clinic. If you start to do this on a larger scale, could you really start looking, and especially when you start looking over time, could you really start looking at some of these communities as fingerprints or different predictors of things? And again, remember, women who don't have BV are dominated by Lactobacillus inners or crispatus. The other thing that's in this paper, which I think is also very important and draws the link between just describing the presence of those bugs and the endpoints of symptoms, or in this case, the AMSL criteria is shown here. This simplicity of this slide belies a very sophisticated analysis where Sujatha and her team of computational biologists and statisticians went in to look at what bacterial taxa were associated with the individual AMSL criteria. And it turns out that the really big players here that affected all components of the AMSL criteria, which are abnormal pH, clue cells, an amine odor, and vaginal discharge, are Gardnerella vaginalis, Leptotrichia amnionii, again, getting back to that Smithia leptotrichia group, Egerthella, and Adipobium vaginae. So, and those are going to be critical, so keep those in mind. And I think this is the kind of thing we need to start doing on a more sophisticated basis. So my conclusion from this body of work, not just Sujata's, but everybody's that's been working in this area, is that all BV is consistent in having elevated pH and loss of non-inners lactobacilli, meaning Crispatus and Jensenii, which likely accounts for the consistency of adverse reproductive and sexual health outcomes. So to me, that's a common pathway, right? The environment is disrupted and it's got those things. Otherwise, I think that there are probably discrete types of BV, and this is totally a hypothesis on my part, although I'm not the only person who has thought about this, that may be defined by community composition of individual species with direct impact on local immune environment. And I would call that an individual pathway. And that may explain, not just because of the microbes involved, but also because of the host response, why not everybody with BV has the sequelae that we think about. So I think that's worth thinking about. So I want to shift a little bit, and I want to come back to Kevin's talk last night. This is a group that's the World STI HIV Congress, so I want to shift to talk a lot about much of the talk about this relationship with HIV, because I think this is a perfect lens to start looking at this diversity question and the host inflammatory response. So just a reminder, this is an old meta-analysis, but I still really like it. I think it was done well, and it's often quoted from Adeshili et al. In 2006, I think now. And I think it's been borne out by many subsequent smaller analyses. And the consensus is that women, again with BV, just let's use that term with the caveats I mentioned, have about a 60% increased risk of acquiring HIV infection through vaginal sex. And that's if you control for everything else, STIs, types of and frequency of sexual behavior, not controlling for partner's viral load. What's missing in all these analyses is the male partner, which I think is a huge weakness, and we have not really come to terms with that. But I think just across the board, you can use this 50 to 60% increase as a pretty robust example. Many of you may not recall that Craig Cohen from the Partners in Prevention study also published a very nice paper in PLOS Medicine a few years ago showing that women with BV were also more likely to transmit HIV to their male partner. And this was great because there actually was a biological gradient with increasing abnormality of the vaginal microbiome. So as your Nugent score got more and more abnormal, the risk of a woman with HIV transmitting HIV to her partner actually went up to the point of, as you can see, about 7-fold. They tried very hard to make this association go away. It's a beautiful epidemiologic analysis for those of you who want to try to understand methodologically how complex this is. They controlled for everything. I'm hiding it there by my summary box, but they really tried to control for everything, and they did not make it go away. What's the main hypothesis that would have explained this? That the women with BV had higher rates of genital shedding or higher genital HIV load, and that explained it. Even when they controlled for that, they couldn't make this go away. So it's still something else that's going on, okay? I also like to refer back to this really nice analysis that I think hasn't gotten enough attention by Lynette Masese with Scott McClellan and their group. And what they did was to revisit data from the Mombasa cohort that University of Washington research staff have been following for over 20 years now, actually closer to 30, I believe. And what Lynette did was to look at 20 years of data and look at HIV acquisition in this cohort of women and attribute the increased risk of HIV acquisition to the conditions that these women experienced over time. Incredibly unique opportunity to look at a database with the same women over 2 decades where vaginal infections and STDs were monitored throughout. What did she find was, regardless of the time period, you can see they're broken out by 4-year periods there. Of course, genital herpes, right? Acquisition or prevalence of genital herpes had by far the highest population attributable risk percent. So, of course, we can't forget that. But the next most important thing was bacterial vaginosis. And again, that accounted for about 20% over time. Absolutely no difference over that time period. So when you think about the prevalence of BV in these women and you think about the incidence of HIV acquisition, you're talking about an incredibly important attributable risk percent. So worth taking a look at that. How does this happen? Well, this slide I put together a while ago, and I update it pretty frequently, although there's now so much data that I can't really put everything on it. I'll just try to sort of point out that there are some general principles that people have postulated mediate this increased risk of HIV. Clearly, you lose your hydrogen peroxide production by your good lactobacilli. Hydrogen peroxide is directly virucidal. Alkaline pH actually can directly activate CD4 cells. Also, you can get activation of Langerhans and CD4 cells directly by these bacteria. And then the cytokine story. Now, the cytokine story could be related to all of this, but there are really interesting accruing data. We've always known that cytokines are disrupted with BV, but it does appear that some of these really promote local HIV replication and increased shedding. And there are a number of studies there that you can take a look at. Rupert Kahl's group very early on showed that successful BV treatment actually reduces some of these disrupted cytokine and decreases activated CD4 T cells at the endocervix, which is really very interesting. It's been tough to replicate that. So we'll talk about that in a second. Let me see if I can maybe go back. I had some trouble last night. Okay, so that is just some idea about what's going on. I would say where we're at right now is that we finally have some new data to help us understand this mechanistic link. It begins with inflammation, and I put that in quotes because everybody has a different definition of what inflammation is. And it ends in some confusion, which is how I'm going to probably end this. But it is a start, and I think it's a really exciting thing for us to start thinking about. So this is an older paper, too, by Andrea Thurman at Conrad with a number of colleagues. But it's a nice paper if you haven't really started to delve into this concept of how BV modifies the local immunology. One of the key things I think is important is that IL-8 is increased. There is production of sialidase, which actually destroys IgA, and there is IgA in the vagina, and in fact, there is IgA specific to Gardnerella vaginalis. Sabina Cacci a long time ago showed this. So again, all of these threads coming together. So many ways that you could conceivably postulate that this environment would be very conducive to BV. So we've known all of this for a long time, but again, putting this together with real-time HIV acquisition studies has not really happened. What I think is new since those papers are published is that we've started to get into an era where we're using functional assays, and I'll describe that in a moment, as well as omics. And by omics, I mean metabolomics, proteomics, that sort of thing, to really start looking at this a little bit more carefully. Betsy Harold's group at Einstein has done some very nice work using an assay that looks at activity of vaginal secretions, vaginal fluid, against E. coli. It's not really clear what confers this E. coli effect, but it seems to associate very strongly with healthy features of the healthy vaginal microbiome. Lots of work coming out on that. The other thing that group has shown is that inhibitory activity is associated, when you use proteomics, with proteins produced by L. crispatus and L. jensenii. So I think it's very likely that these bacteria are producing something that is actually promoting this healthy and protective effect. Later data from the Rupert Kahl paper presented at the HIV Research for Prevention meeting in 2016 again emphasized that when you've got these cytokines associated with BV, you not only increase your endocervical CD4 T cells, which increases the target for HIV, but you also, when you use these new proteomic approaches, get evidence of epithelial disruption. So you put this all together, you've lost your hydrogen peroxide, You are not producing these proteins that seem to be associated with protection or at least functional anti-E. coli activity, which has been associated with vaginal health, and you may be disrupting the vaginal epithelium at a very, very microscopic level that we can't really see. It's not like a vaginal ulcer. I think this is starting to put together the picture that we might be trying to tell ourselves about why this is happening. And I would refer you to a nice review paper by Rupert that talks about this, that again is a little bit of 2015, but really goes into some of these issues in detail. Now, people got really interested in this last summer, a year ago, because at the Durban IAS meeting, the CAPRISA group, as well as Scott McClellan's group from University of Washington, presented some fascinating data. And this is where I would say people started finally to remember that HIV is acquired in the vagina. Isn't that like amazing? The virologists were just like, wow, really? I can tell you a funny story about that later. So what did the CAPRISA group present that was so interesting? And this data has finally just been published. Nikki Klatt published it in Science just June 2nd, I believe, and I'm going to show you some slides for that. This group had shown previously, that's the Melisana JID paper from 2012, that the women in the CAPRISA study, and for those of you who don't remember, CAPRISA was a study of pericoital tenofovir gel versus placebo gel for prevention of HIV acquisition, and it showed a reduction of HIV infection in the gel users by about 50%, and in the highly adherent users about 68%. So it actually worked in that study, much to many people's surprise. That group had generated what they called was an inflammatory profile, and the top picture, which you're not supposed to see, but just a reminder for me to say it, is that they had measured a large panel of cytokines and defined a group of cytokines that they were confident defined an inflammatory environment, and that inflammatory environment by itself was associated with HIV. And that's what this graph shows you, or this table shows you. The women who had genital inflammation, again, is defined by, I would say, a rigorous file, and the top had an odds ratio of 3. Is that surprising? Not really. We've known for a long time that having an STI, right, particularly trick, or an inflammatory, even candida, actually vaginal, is a risk factor for acquisition of HIV. But interesting to see this with a very sophisticated cytokine analysis in a group of young women in sub-Saharan Africa. So where did they go with that from there? What they then did, which is new, is with the help of Adam Bergener's group in Manitoba, using proteomics, not using 16S, ribosomal DNA analysis, sequencing, or anything like that, and I'm going to come back to that issue. They then went in to look at what the microbiome was that was associated with this inflammatory score. And what you're looking at here is horizontally a spectrum of community types. So remember the paper that I showed you before with Sujatha's nice graphic where all the community type, the diverse communities associated with BV were at the top, the more homogeneous communities were at the bottom. This is just a horizontal version of that. The women on the right with all the colors are getting into increasingly diverse community states, okay? And you can see here that the community cluster or community state type 4 was linked most strongly with this genital inflammation and HIV. And that was, again, using this proteomic definition of the microbiome, okay? What was in that cluster when they looked more carefully, again, not by specific PCR, but by this proteomic approach, was Prevotella bivia. Prevotella is one of those big bad anaerobes of BV. If you go back and look at the old prospective data that Sharon Hillier, Dave Eschenbach, others did of adverse outcomes in pregnancy, Prevotella is one of those black anaerobic gram-negative rods that was associated with worse outcomes of pregnancy. But fascinating, here it is again, 25, 30, 35 years later, coming up in this proteomic analysis. And what they showed was that P. bivia was strongly associated with genital inflammation and HIV acquisition. So if you had P. bivia in your vagina, nothing about quantity, nothing about PCR specificity, all caveats. And very small numbers, so keep that in mind, please. 19 times more likely to have genital inflammation and 13 times more likely to acquire HIV. Now, this is a lesson in interpreting these data with caution. What was the initial reaction to this? Oh, we need to find antibiotics that will kill Prevotella bivia and start infusing them into the vagina continuously. That is not the appropriate reaction, right? You really have to take a step back and try to think about what is going on here. Is this a marker for something? Do we need to look at relative quantities? Is there a community associated with it? Are there behaviors that are associated with this that are actually the true mediator of this phenomena? No idea. So lots of, frankly, scientifically, I think, irresponsible knee-jerk reactions to these data were evident. So at the same meeting, that got a lot of press, a lot of reaction. At the same meeting, Scott McClellan and colleagues, as I mentioned, of the prior Mombasa data, showed a very similar analysis, which was not a talk, was a poster. And what they did was to gather data from a large cohort of women from several settings in Africa. Many of them were pregnant. I think about a fifth of them may have been in pregnancy studies. And they did a very careful statistical case control, nested case control analysis that Sujatha actually led the laboratory work on to look at a very similar question. When you look at the vaginal microbiota in these women, prior to HIV acquisition, can you look at cases who acquired HIV versus controls who did not and identify some signature disruption in those cases that could explain why these women got HIV? So this is what it looked like, and they characterized the microbiota by deep sequencing and qPCR. So very different approach. What did they find? Well, not surprisingly, the cases versus the controls had more diversity, right, in their vaginal microbiome. Again, that's really, to me, not very different than saying BV is a risk factor for HIV. That's really what we've been saying all along. And I should mention this has not been published yet. It just was presented. So this is just the diversity index in the cases on the right versus the controls on the left. And you can see the individual bacteria there that were looked at in this analysis. When they went in there with specific quantitative PCRs, so now getting much more refined than the previous analyses, to look at whether you could find a predictor by quantity of those vaginal bacteria, you can see that they did find some interesting trends. Egerthella, remember, which was in that confluence of the AMSL criteria, came up actually as a risk with a significant p-value. A couple of others there, including our friend Leptotrichia snethia, not surprisingly maybe, because we know that's associated with urethritis, and there's going to be a nice presentation on this, I believe, today. And then Mycoplasma hominis, not just barely significant. So this did not show Prevotella. They looked very specifically for that. So what does that mean? Why is it different? What can we say? You have one group saying that we need to go in and target this bacteria and maybe even develop a vaccine for it. And then another group that's saying we really don't know what's going on here. Well, I think there are a couple of different reasons. And again, this is why I would really urge caution, and when I come back to this at the end, talk about where we need to go from here. Very different populations of women. There are lots of unmeasured variables, and as I said, we have no data on exposure to male partners' microbiome. And I think until you put that into this equation, talking about sort of drastic modifications to the vaginal microbiome is not particularly thoughtful. But let's come back to that. As I mentioned, they use different techniques to define the microbiome, and this is part of the evolving scientific landscape, right? The traditional, now traditional, it's traditional since not very long ago, method is really the deep sequencing, and that's what Scott and his group did to direct and define quantitative PCRs to look at these bacteria. The Caprisa group used this proteomic approach. What does that mean? They basically search for bacterial peptides, so proteomics peptides, that were then associated with a database derived from earlier 16S ribosomal RNA work. Okay, so it's derivative. It's not wrong. It's not less robust. It just is a different approach, and you got to understand the limitations of it. And again, There wasn't specific quantitative PCR. They estimated bacterial abundance by summing these protein spectral counts, which is very creative, but I think needs more validation before we really sort of go to say that this is what's going on. Now, on the heels of this fascinating paper that I think you should know about if you haven't seen this, by Doug Kwan's group, this is Christina Gossman's paper that came out just earlier, I want to say, let's see, what are we, 2017? It's earlier this year, February of 2017. And this is an interesting paper because it took advantage of something called the FRESH cohort, also in South Africa. But what's interesting about the FRESH cohort is that it is very young women, teenagers, essentially. I think maybe up to 21. And I'm going to tell you what they did here. They looked at 236 young women, and they defined 4 vaginal community types at baseline using more traditional, quote-unquote, sequencing methods, as I mentioned. And again, this is not surprising, right? If you look at these community state types, this is pretty much what I showed you before. You've got type 1, which is dominated by Lactobacillus crispatus in orange, type 2 in green, that pesky Lactobacillus inners, and then increasingly diverse, some would say disrupted, vaginal microbiota to the point where you go all the way to this community state type 4, okay? So keep that in mind. I do want to point out in the community state type 4, that sort of mauve color that's dominating in the middle range of the drawing, that is Prevotella species. So it is there, and it's going to come back to haunt us, okay? What they did then was to assess whether these community baseline types predicted acquisition of HIV in just 31 women, okay? Only 31 women who are cases. So again, caveat. They sequenced the V4 region, and they also measured immune mediators, and then novel, they did an associated animal model assessment in the mouse, and that's what I want to show you. There's a couple of really fascinating findings from this. They looked at chlamydia, they stratified by chlamydia. They didn't really, chlamydia was associated with an increased risk of HIV acquisition, actually a 4-fold risk. So for those of us who are still interested in chlamydia, that's a pretty important finding. But what I want to show you is the small graphic in B. And this breaks down the HIV acquisition by those community state types. There are a couple of stunning things to me in this very small table. Again, small numbers. No one who had community state type acquired HIV in this study. The incidence was 10%, I think, 8 to 10%. I can't remember what it was, but it was very, very high. It's 31 women out of 200, so that's what it was. None of those women acquired HIV. 112, really, really impressive. Of the group of community type 2, the inners group, 9. So this, I think, again, tells you that inners is not really helping here. What's it doing, and why is it associated with a normal clinical environment? We don't really know. Look at the acquisitions in types 3 and 4, particularly type 4. Really, almost all of the acquisitions occurred in 3 and 4, but nobody in 1. So I think that's really, really very, very telling. And the other things that they showed in assessing the immune environment in those women was that when you had this community type 4, and again, getting more sophisticated from the old sort of just straightforward cytokine analyses, was that they had more HIV target cells in tissue. They had elevated levels of MIP1 alpha, MIP1 beta. These are CCR5 attractants. So you're, again, attracting the target cells. Not surprisingly, our old friend IL-8, IL-1 alpha, TNF alpha, IL-10, and also IL-17, which is a newer sort of dimension. So really nice, I would say, work. The last thing that they did, which was kind of cool, and that's just the first 3 points are just some summaries, is that they took Prevotella. This was sort of separate, but they were going down that Prevotella road, again, because of what Caprisa had shown, and again, because of the Prevotella dominance in community state type 4, and they gave it to mice, and they put it into the vagina of mice. Mice are not necessarily a great model for the human vagina, but they will do. That's one of the problems, of course. We don't have an animal model of BV, which is something I think would be really lovely. And they actually showed that when you did that, you increased activated genital CD4 cells in the mice at the cervix. So this story is coming together. It's kind of ragged, Lots of pieces that involve mice and small numbers and different approaches, but I think it is becoming a cohesive story. Now I'm going to shift just a tiny bit, and then I'll wrap up by, actually I hope, creating a transition for Sinead to come up and talk about PrEP. Everybody knows that the data on PrEP are fantastic for men. We're already seeing declines, Washington, D.C., probably in part because of treatment as prevention, but also maybe PrEP is seeing dramatic reductions in the incidence of HIV. Great data for men, not such great data for women. And why do we think that happened? We think women are spacey and they don't adhere to PrEP, and I can say that because I'm a woman. Is that really the case? Okay, we have blamed the lack of PrEP efficacy, both oral and vaginal, with tenofovir-based products, okay, on adherence, and even the dapivirine vaginal ring. We've blamed the less effective finding in young women on low adherence. So I think the question is now, is it all non-adherence? Clearly in the study that I was involved in the FemPrep study, and in the FAX-001 study that Sinead was involved with vaginal tenofovir, adherence was a huge problem, and it did mediate a lot of the lack of efficacy. But there are some very intriguing data coming out. First of all, we know that the rectum is not the vagina, in case you didn't know that. I'm happy to tell you that definitively. I don't have imposter syndrome when I say that. But it's less, part of the challenge is that it probably metabolizes tenofovir differently. So tenofovir is simply not as activated and concentrated in endocervical T cells or endocervical cells as it is in the rectum. So again, a biological disadvantage from women, and that's the Cottrell paper, which I can talk about separately. I didn't want to take time. So does the vaginal environment play a role? Well, this is Nikki Klatt's paper, which just came out in Science literally a couple of weeks ago. And what this is the publication that followed on the Durban IAS meeting. And they, again, used bacterial profiling by proteomics or mass spectrometry in the 688 women from the Caprisa 004 trial. And again, you're seeing here that distribution of the community. And they then went in, again, to take a look at the cumulative HIV infection probability by treatment assignment in women with vaginal lacto versus non-lacto bacterial dominance. And what you can see here is that the probability of HIV infection in the placebos versus the controls in the lactobacillus dominant group on the left did separate out. It was clearly different. So tenofovir worked in the women who were lactobacillus dominant. In the women who were non-lactobacillus dominant, tenofovir did not work at all. Now, what's the problem with this? To me, the biggest problem is lactobacillus dominant just lumps everybody together. So there is no distinction between inners and crispatus. And when you think about Doug Kwan's paper that showed that dramatic difference in HIV incidence for community state type 1 versus 2, I think that's a big problem. Nonetheless, they still showed this marked distinction in tenofovir efficacy by whether these women had lactobacillus or no lactobacillus. And that, I think, was pretty compelling, okay? They then went a little bit further and looked at the metabolism of tenofovir by Gardnerella vaginalis and BV-associated bacteria. This has gotten a lot of press. The bottom line, I won't take you through these figures in the interest of time, but the bottom line is that they added Gardnerella vaginalis, Adipobium vaginae, and Lactobacillus crispatus, and I think Lactobacillus inners, if I'm not mistaken, yes, to in vitro cultures systems of Gardnerella vaginalis, and essentially showed that with tenofovir in place, the metabolites of tenofovir were effectively gone, or tenofovir, functional tenofovir, was taken out when Gardnerella was there. So this has given rise to this concept that Gardnerella vaginalis essentially chews up tenofovir, and maybe the women in trial who were lactobacillus non-dominant had a lot of Gardnerella, and that was chewing up whatever tenofovir they were using. Maybe they weren't that adherent, so even if they had been protected through partial adherence, then they were out of luck because their Gardnerella was the bad boy, and it is a bad boy. So what does that really mean? How should we put this together? Well, I think you can say from these data, fascinatingly, that tenofovir gel efficacy was threefold higher in women with this non with lactobacillus dominance and not effective in those who did not have lactobacillus and were dominated by Gardnerella. So I think the vaginal microbiome probably interacts with some topical antiretrovirals. We don't know what that really means or what that's about. I think the Klatt paper has some great hypotheses, but there could be other things going on here too. I already mentioned the PK/PD, pharmacokinetics, pharmacodynamics of the rectum versus the cervicovaginal environment. Unfortunately, that's different. However, beautiful data from the Partners in Prevention study that Renee Heffron presented at CROI, saying it really doesn't matter if you take oral PrEP. So you should be fine. If you're adherent to your oral PrEP, you're going to overcome these problems. At least that's what they saw. Nikki Klatt did finish her paper, though, by saying vaginal pH screening and microbiota monitoring and treatment are potential adjunct interventions to enhance PrEP impact in women. Well, what are we going to do? We're going to screen and monitor, and what? Are we going to treat them? Are we going to give them lactobacillus? That wasn't really addressed. So I'll just point out a really, I think, promising approach. This is Jen Balkus's paper, again from last year, that presented the data from the Preventing Vaginal Infections study. And if you may recall, Scott McClellan's group showed previously that giving women periodic presumptive treatment for vaginitis, namely monthly metronidazole and an antifungal cream, reduced, not surprisingly, the incidence of vaginal candidiasis and BV. What this group then went on to do was to look at whether that reduction in BV was associated with a reduced risk of acquisition of sexually transmitted infections. So let's put our hypothesis to the test, right? And indeed, small numbers, but what Jen and colleagues showed was that if you combine the outcomes of chlamydia, gonorrhea, and mycoplasma genitalium, you showed a significant reduction in acquisition of STIs. Small sample size, but clearly a very interesting trend that should really, I think, re-energize those of us who are working hard and care about maintaining a normal vaginal environment. So I am basically finished with 35 seconds left. So just to wrap this up, how would I conclude? I think clearly a Lactobacillus crispatus dominant microbiome is associated with a lot of good things, and especially a lower prevalence and incidence of BV. I do think we need a lot more work in this area. We really are just starting to pull these threads together, and I think maintaining this environment, the normal environment, is really going to be the holy grail. But we desperately need new interventions, right? I mean, what do we have going on? I love this cartoon. Actually, I got some pretty good ideas when I was in the box. The box is a safe place, especially when you're talking about vaginal microbiome. It's a safe place, right? We want to think inside the box. We want to think about new antibiotics. Maybe we want to do a new version of metronidazole, right? Maybe we want to do a new version of whatever. I don't think so. What do we need to do? We can't solve our problems with the same thinking we used when we created them. Kevin DeCock said this last night, using different words. And I think when we think about what we should do with the vaginal microbiome, there are a host of things we need to start being more creative about. I think creative approaches to partner management, Kat Bradshaw and I have talked a lot about doing a study involving topical and oral therapy for males. I think not having topical therapy probably just doesn't make much sense to me. Modifying the hormonal environment. There's lots of ways we could do that. We're doing a study now in Kenya, and Ken Nguri is here, going to give you an update on our study looking at NuvaRing to modify the vaginal environment in Kenya. Lactobacillus repletion, I mentioned Craig Cohen's study. Vaginal hygiene interventions, beautiful summary by Nicola and colleagues looking at the risk for vaginal hygiene, incredibly prevalent. Biofilm disruption, new antimicrobials, and menstrual suppression. Menses is very much associated with onset of BV, and we could certainly deal with that with hormonal therapy. So I'll just stop there. I really want to thank IUSTI for this incredible honor, especially David, who's just been a wonderful, wonderful colleague over the years, and then a lot of people who've given me slides or really talked through some of these issues with me. And I'll stop there, and thank you very much for listening.